Assalamualaikum and good day. We are from Group 16 and today we will present our assigned article titled Whole Genome Sequencing Analysis Identifies Sex Difference of Familial Pattern Contributing to Phenotypic Diversity in Autism. My name is Noakila Batrisha binti Muhammad Hafiz and my metric numbers is A203878. I will explain the introduction part of this study. The background of this study focuses on sex difference in autism. Autism is more common in males than females, with a prevalent ratio of around 4 to 1. This suggests females may require a higher genetic risk before developing autism symptoms. Previous study found that females with autism carry more de novo protein truncating variant or de novo PTVs. De novo variants are new DNA changes that occur in child and are not inherited from the parents, while PTVs can produce incomplete protein that may affect brain development and contributing to autism. Intellectual disability is also strongly linked to this harmful mutation and reduce the male to female autism ratio from about 4 to 1 to 3 to 1. However, most previous whole genome sequencing studies focuses mainly on European population, leaving East Asian population underrepresented. Next, based on these gaps, the study was conducted for several important reasons. First, researchers aimed to better understand differences in autism characteristics between males and females because sex may influence intellectual disability, autism symptoms, and clinical diagnosis. Second, family-based analysis was used to separate inherited genetic risk from new mutation by studying siblings and parents who share genetic background but may carry different genetic burdens. Finally, since previous study mainly involved European population, this East Asian genome study allows comparison across ancestries to determine whether autism genetic patterns are similar or different between populations. Therefore, this study had three main objectives. First, to examine sex difference in genetic burden by comparing the Korean autism cohorts with European cohorts such as SSC and SPARK. Second, to perform phenotype analysis in unaffected family members, including siblings and parents, to understand whether females may have greater tolerance to inherited genetic risk. Third, to investigate how intellectual disability and autism symptom severity influence sex difference in genetic burden between males and females.
A very good day, everyone. My name is Neha Vigneshwaran, metric number A203827, and today I will explain the methodology used in this study on sex differences in autism using whole genome sequencing analysis. First, the researchers recruited participants from several cohorts. The main cohort consisted of Korean autism families involving 673 families and 2,255 individuals. They also included the COGAS Korean control population and comparison cohorts from the SSC and SPARK databases to strengthen the reliability of the findings. Next, clinical data were collected from participants. These included autism symptom severity, social communication abilities, repetitive behaviors, IQ scores, adaptive functioning, and family-based phenotype assessment. These measures allowed researchers to compare both genetic and behavioral differences between males and females. After that, DNA samples were collected from whole blood and underwent whole genome sequencing, or WGS, using Illumina sequencing platforms with approximately 30 times sequencing depth. This enabled researchers to analyze the entire genome in high detail. The sequencing data went through data processing and quality control. During this step, low-quality samples and unreliable variants were removed, while sex and ancestry verification were also performed to ensure accurate analysis. The next stage involved identifying important genetic variants, including de novo variants, protein truncating variants, or PTVs, and missense variants. These variants are strongly associated with autism risk. Following this, the variants were annotated and classified using bioinformatics tools such as VEP, Lofty, MPC, and LEUF scoring system. These tools help prioritize potentially harmful genetic mutation. Researchers then calculated phylogenetic scores, or PS, to estimate inherited autism risk across individuals using GWAS reference data. Finally, statistical analysis such as t-test, ANOVA regression, and gene phenotype association analysis were performed to compare males and females, autism cases and siblings, and different family groups. Overall, this methodology allowed researchers to investigate how genetic burden and autism-related traits differ according to sex and family patterns. Thank you.
My name is Nur Shalina binti Muhammad Shahjahan, A203695. Now I will explain the results of this study. First, for de novo protein truncating variant or PTV, the study found that autistic females carry a higher burden compared to autistic male. In the Korean cohort, females had 0.065 variant per person, while male had 0.033. In SSC, females had 0.103, while males had 0.065. In Spark, female also show a slightly higher value 0.062 compared to 0.050 in males. This suggests that females with autism may have a higher genetic liability threshold, meaning a greater genetic burden may be needed before autism traits are clinically expressed. Second, for polygenic burden, autism cases show higher polygenic score than non-autistic siblings. This means inherited common genetic variants also contribute to autism risk. However, sex differences were not consistent across cohort. In the current cohort, males had higher polygenic score than females, but in SSC and SPARC, the pattern was different or not significant. Third, the study found that intellectual disability influenced sex differences in genetic burden. Autism cases with intellectual disability had a higher de novo PTV burden than cases without intellectual disability. For example, in the SSC cohort, autistic females with ID showed the highest burden around 15%, while males were around 7%. This showed that ID is strongly linked with damaging de novo variant. The sibling analysis also show an important family pattern. Male siblings of autistic females had the most severe social communication difficulties. In the slide, they show the highest social communication score, 1.25, compared with the other sibling groups. This suggests that males may be more affected by inherited autism-related genetic risks. Lastly, for parental burden, mothers show higher polygenic burden than fathers. In the Korean cohort, mothers had about 0.052 SD higher polygenic score than fathers. And in SSC, the difference was stronger at 0.105 SD. However, mothers still show less severe symptoms. This supports the idea that females may tolerate genetic risk better than males. Overall, the results show that autism genetic liability is influenced by both sex and phenotype, especially intellectual disability, social communication traits, and family genetic pattern.
This study makes an important contribution to autism genetic research, especially in non-European population. It improves our understanding of sex differences, genetic liability, and phenotypic diversity in autism. The family-based analysis also provides stronger evidence for the female protective effect, where females may tolerate higher genetic burden before showing autism symptoms. However, there are also some limitations. The study relied on European-derived polygenic scores, which may be less accurate for East Asian population. In addition, female sample size were still limited and environmental or epigenetic factors were not deeply explored. For future studies, larger multi-ethnic genomic research is needed together with better autism diagnosis in females and further investigation into female-specific protective mechanism and environmental influences. In conclusion, this study supports a sex differential liability model in autism, where comorbid intellectual disability (ID) and overall symptom severity contribute to differences in genetic burden between males and females. Females were found to tolerate a higher inherited genetic risk while showing milder clinical features compared to males, suggesting a higher liability threshold in females. Overall, these findings emphasize the importance of considering phenotypic heterogeneity and family-based approaches to better understand sex differences in autism. That's all from our group. Thank you.